heart. In short, we discuss about how the neural regulation is there in case of the heart. If you know, suppose this is the structure of the sinus venosus. These are the atria, and there is a presence of this ventricle, and this is a spinal cord, and there is a presence of the vagosympathetic trunk. This vagosympathetic trunk contains the sympathetic as well as the parasympathetic fibers. Parasympathetic fibers in the presence in the form of the vagus nerve, and the sympathetic fibers present as a sympathetic fibers. While there is a presence of the sympathetic chain of ganglion, there is a presence of sympathetic chain of ganglion. This sympathetic chain of ganglion, the fibers which will originate from the spinal cord, they will enter. So this is a sympathetic stellate. These are the stellate ganglions. This is the sympathetic chain of the ganglion, and this is a spinal. The fibers which will originate and terminate on this ganglion. Now these are the preganglionic sympathetic fibers. These are the preganglionic sympathetic fibers. Okay. Now there occurs the formation of the synapse in this ganglion, and as a result, there is a origination of the second order neuron. These are called as a post-sympathetic fibers. These are the post-sympathetic fibers. And from here, from the skull, there is again origination of the parasympathetic fibers in the form of the vagus nerve. Now the fibers from the skull, they will come out from the skull, and they will join these sympathetic fibers. The proportion of this vagus fiber, of the parasympathetic fiber, is more, and structure they form that is called as a vagosympathetic trunk. That is called as a vagosympathetic trunk. Now this is a vagosympathetic trunk which will contain the mixed type of the nerve fiber. This is called as actually vagosympathetic trunk. Now this vagosympathetic trunk, these are the fibers of the parasympathetic nervous system. These are the preganglionic fibers. Preganglionic parasympathetic fibers. These are the preganglionic parasympathetic fibers. In this vagosympathetic trunk, if you check its percentage is about 75%. In the vagosympathetic trunk, there is a 75% of parasympathetic fibers. These parasympathetic fibers are present in the form of the vagus nerve. Which is a inhibitory. Okay. Why? It contains only 25% of the sympathetic fibers. It only contains the 25% of the sympathetic fibers. Now, if we discuss about these are the preganglionic sympathetic fibers. Second order neuron will form, so it will contains this now contains vagosympathetic trunk contains the preganglionic parasympathetic fibers and post-ganglionic sympathetic fibers. Now, and these are supplied to the heart. This is a sinus venosus. These are the atria, and this is a single ventricle. Okay, but there is a presence of the different ganglion. There is a presence of this different ganglion. Ganglion is nothing but the collection of the nerve cell outside the CNS. Is it clear? and these fibers, some fibers will terminate on this ganglion, some fibers will terminate on this ganglion, and some fibers will supply to the third ganglion. This is the Ludwig's ganglion. This is a Ludwig's ganglion. This is the Wider's ganglion. This is third one is the Rimax ganglion. And what happens? What happens when you stimulate the? If you compare the percentage of the in the vagus trunk, vagosympathetic trunk, if you compare the percentage, what is the percentage? 75% is inhibitory fibers in the form of the vagus, that is the parasympathetic fibers, and 25% are the sympathetic fibers. Now, if you stimulate this now, vagosympathetic trunk, if you stimulate this vagosympathetic trunk, as the proportion of the inhibitory fiber is more, so it will generate the inhibitory effects on the heart. It will generate the inhibitory effects on the heart. Now, what happens when you stimulate the vagosympathetic trunk? When there is a stimulation of the preganglionic sympathetic fibers, it releases acetylcholine. It releases acetylcholine. Okay. When there is a stimulation to the preganglionic parasympathetic fibers, it will come up to this ganglion. These are the preganglionic parasympathetic fibers. Is it clear? At the ganglion level, they will release ACH, acetylcholine. Okay? Understood? And when there is a release of this stimulation of the vagosympathetic trunk, where this parasympathetic fibers releases the acetylcholine, while these post-ganglionic sympathetic fibers, they will releases the adrenaline. They will releases the adrenaline. So we can look at such a way. Pre-ganglionic parasympathetic fiber. What they release? Pre-ganglionic parasympathetic fibers, they releases the ACH. Pre-ganglionic sympathetic fibers, they releases the ACH. Okay. Then post-ganglionic means the pre-ganglionic fibers, post-ganglionic parasympathetic fibers. That will discuss after. As there is a stimulation of the vagosympathetic trunk, this vagosympathetic trunk contains the pre-ganglionic parasympathetic fibers. On stimulation, it releases the acetylcholine at this level, at this middle ganglion. Is it clear? Simultaneously, there is a stimulation of the sympathetic fiber also, which are the post-ganglionic sympathetic fibers. They releases the post-ganglionic sympathetic fibers. They releases the noradrenaline. They releases the noradrenaline. Is it clear? Then post-ganglionic parasympathetic fibers, they releases the ACH. They releases the ACH. Now, what happens when there is a vagus stimulation? Effect is inhibited. Heart stops in task. Heart stops in task. And there is a release of the both neurotransmitter. As it contains, vagosympathetic trunk contains the preganglionic parasympathetic fibers. At their terminal end, they releases the acetylcholine. Effect is inhibitory. Number of the fiber is more, and there is a release of the, again there is a stimulation of the post-ganglionic sympathetic fibers. Again there is a release of the noradrenaline. Effect is stimulatory, but if you compare the number of the fibers, so the number of the neurotransmitter released by the parasympathetic fiber is always more. So here the parasympathetic system is dominant, and they possess the, when there is a stimulation, they possess the release of the acetylcholine in higher quantity and possesses heart stops in task. Is it clear? Now what happens at this junction? This is called as a crescent line. This is called as a crescent line. This crescent line actually is a demarcation between the sinus venosus and the atrium. It is actually demarcation between the sinus venosus and atrium. But if you stimulate this part, then what happens? Post-ganglionic parasympathetic fibers get stimulated. Post-ganglionic parasympathetic fibers get stimulated. Is it clear? Again, there will occur the stoppage of the heart in a diastole. Stoppage of the heart in a diastole. So, have you understood? When there is a stimulation of the vagosympathetic trunk, vagosympathetic trunk contains the two types of the fibers. Percentage of the parasympathetic fiber is 75%. Sympathetic fiber is only 25%. It contains the pre-ganglionic parasympathetic fibers in 75% and only 25% post-ganglionic sympathetic fibers. Is it clear? So, when there is a vagus stimulation, heart stops in diastole. But when there is a crescent stimulation, again there is a stimulation of the post-ganglionic parasympathetic fibers. So, again heart stops in a diastole. So, have you understood this? So, through this, just you want to collect the information that is stimulation of the pre-ganglionic parasympathetic fibers releases the acetylcholine. Pre-ganglionic sympathetic fibers releases the acetylcholine. Post-ganglionic sympathetic fiber releases the noradrenaline. It is a stimulant. And post-ganglionic parasympathetic fibers releases the acetylcholine, which is a inhibitor. Is it clear? Now we are going to discuss about the effect of these two drugs, that is the adrenaline and acetylcholine on the heart. Is it clear? Have you understood about this? Yes or no? Now, for this experiment, we are going to use this myogram board, this Sherrington revolving drum. This is a induction coil. Now, this is actually movable electrode that we have seen that there is a presence of the white crescentic line. We will take the connection from this secondary coil and we will stimulate this crescent with the help of this movable electrode. Is it clear? Have you seen the normal cardiogram? Yes or no? And recording, we will record this tracing of the normal cardiogram on this revolving drum. So what will be the record? This record will be in this form. But what happens actually? How this record is? What is the systole and what is the diastole? So that we are going to discuss. Now, see that with the sinus vena, this is a ventricle of the heart. These are the atria, and then there is a presence of the sinus venosus. Then we are going to pass a pin through this lower portion base of the ventricle. We are going to take the care that we are going to pass this pin only with through the muscular mass only. We have to take the precaution there should not be puncture of the ventricle. Is it clear? So suppose there is this pin is there in this form, and this pin is tied to by the thread to the liver. So the liver will be in this form. Understood? This is a stand. This is actually Sherrington Starling liver actually, which is a highly sensitive. And what happens when there is a contraction of the heart? What happens to the contraction of the heart? Liver will goes down or up? Liver will goes down. So this will be the liver will goes down. So it will be the systole. It will be the systole. And due to there is a presence during the relaxation of the muscle, and due to there is a presence of this spring, again this lever brought to the normal. So this will be the diastole. So I don't understand this. What is the systole and what is diastole? It is recorded. So if you mount this for by dissection, and if you mount pass the pin through the base of this ventricle, then what will happen? At the end of the ventricle, there will occur the beating of the ventricle or the heart, and there will be recording of that normal cardiac. Okay. Now we are going to discuss about what is the effect of the drug action. If you add this acetylcholine, if you add this adrenaline, what is the effect of this drugs on the normal cardiac? So before that, I want to discuss with you that there is a development of the action potential in case of the SNO. Actually, this whatever the drug we are going to add, this drug has an effects on the three tissues. First is the SNO, second is the conducting system, and third one is the cardiac muscle itself. Is it clear? Now, what happens when there is a development of the action potential? There is a slight slope is there during which the actual only sodium channels will open. Is it clear? And from this point, there is an actual rapid opening of the calcium channel. Long-lasting calcium channel will open. Is it clear? And there is again depolarization. Again stimulation. Firing level, if you reach up to the firing level, again there is a relaxation. So in this form, there is a development of the action potential will occur. This is due to the slow opening. Yes. Sodium channel. Slow opening sodium channel or the leaky potassium. What happens actually during this? There will be this will bring this action potential, resting membrane potential to the firing level. Is it clear? Now what happens at this point? There will occur the opening of the fast opening calcium channels. Is it clear? Fast opening calcium channels. And increase in the calcium concentration, it will be helpful for increase the force of contraction. Is it clear? That we will discuss now actually at a molecular level. Then in some cases, what happens actually when there is a hyperpolarization? Means what? When there is a hyperpolarization, excess potassium goes up. At that time, there should be excess entry of the sodium. There should be, and the time required for this period is more. So when there is a relaxation of the diastole or the contraction, or so as to reach up to the firing level, time required will be more, and that is it is effect of the hyperpolarization. It is effect of the hyperpolarization. Is it clear? Now first we'll discuss about the effect of acetylcholine. So as to find out the site of action of the drug, first we are going to stimulate the vagus, then we will stimulate the crescent. After then we will add the drug, then again we will find out again stimulate the crescent, again we will stimulate the vagus. What is actually meaning? What we have to do this? It is necessary. Why? Before you have discussed before starting this adding this drug, what you have done? You have stimulated the crescent. Means that you have stimulated the vagus. Means that you have stimulated the preganglionic parasympathetic fibers and postganglionic sympathetic fibers. Is it clear? That is, neurotransmitter has been released at the ganglion level, and there occurs the stoppage of the heart in the diastole. Similarly, at the crescent level, when we stimulate the crescent, then again there is an inhibiting neurotransmitter in the form of the acetylcholine will be released, and it will cause the again stoppage of the heart in a diastole. Is it clear? Again, we will add the drug, and we will find out the site of the action of this drug. If you again, then what will be the effect that we will discuss? After that, again, we are going to stimulate the crescent as well as the vagus. So as to find out the site of action of the drug. If the drug acts on the receptors of the ganglion, then receptors of the ganglion will be blocked. Is it clear? After stimulation of the vagus, there should not be stoppage of the heart in the diastole, or after the stimulation of the patient, the heart should not stop in a diastole if there is a blockage of this receptor by the added drug. Is it clear? And if there is a stimulation by or when there is a vagus stimulation and heart stops in the diastole, and if there is stimulation, present stimulation, and again heart stops in the diastole, it indicates that whatever the drug added, it acts on the cardiac muscle. Is it clear? It acts on the cardiac muscle. That we are going to discuss. Now this is a normal cardiogram, and if you stimulate the vagus, if you stimulate the stimulation of vagus, what you are expecting? Heart stops in diastole. Heart stops in diastole. That mechanism we will discuss afterwards. Is it clear? That mechanism we will discuss afterwards. So what it indicates? Heart stops in diastole. Is it clear? After that, what will happen? As you know that there is a presence of the cholinesterase is there. Whatever the excess quantity of the acetylcholine release, this excess quantity of the acetylcholine will be gradually, there will occur the removal of this acetylcholine. So heart will be come out from the inhibitory effect gradually. Heart will be come out from the inhibitory effects gradually, and it shows a staircase phenomenon. It shows the staircase phenomenon in this form. And if there is a complete removal of this acetylcholine, then there is a recording of the normal cardiogram. This is nothing but the staircase phenomenon. Understood why there is a staircase? In the examination, they will ask the question regarding why this gradual increase in the height of contraction it occurs. Okay, the reason is there is a gradual removal of the acetylcholine by the cholinesterase. Again, we will recall. If you again stimulate here the crescent stimulation, again heart stops in diastole. Again, heart will come out from the inhibitory effect gradually. Again, it shows the normal cardiogram. Okay. Now, here we are going to add a drug that is acetylcholine. At this point, acetylcholine is added. As the heart, this acetylcholine has an inhibitory effect. So, what you are expecting? Heart stops in. Yes, what we are expecting? It has an inhibitory effect. Heart stops in diastole. Again, this acetylcholine will be removed by the cholinesterase. So again, there is heart not stops immediately in the diastole. There is a decrease in the rate and the force of contraction. There is a decrease in the rate and the force of contraction because it has an effect on the SL nodals or the pacemaker also, where it will causes there is a decrease in the opening of the calcium channel. Is it clear? Then it also decreases the calcium entry into the cell. Ultimately, first decrease in the calcium entry, so it will decrease the heart rate in the nodal tissue. As it has the effect on the cardiac muscle, decrease the entry of the calcium into the cell. Ultimately, decreases the force of contraction because maximum calcium is available, the maximum force of contraction will be. Is it clear? So what happens? There is a decrease in the heart rate and the force of contraction. So if you want to show the help of this graph, so as to decrease the heart rate, increase the distance between this graph and decrease the height of contraction also. You understood? Decrease rate and force of contraction. This is the effect of the ACH. ACH decrease heart rate, decrease force of contraction. Then again there will be the, if you remove this drug from the site of addition, there will be the normal cardiogram. After that, if you stimulate again the vagus, so the heart will again vagal stimulation at this point. Then the heart stops in diastole. Again, it will come out from the inhibitory effect. If you again stimulate the crescent stimulation, heart stops in diastole. Again, it will come out from the inhibitory effect and shows the normal cardiogram. So what we have done in this experiment, we have stimulated the first vagus. Now recorded the normal cardiogram. First we have stimulated the vagus. As it has inhibitory effects on the heart, heart stops in the diastole. As there is a cholinesterase enzyme is present, it will remove. Gradually there is a removal of the acetylcholine, and as a result, the heart will come out from the inhibitory effects gradually, and it shows the staircase phenomenon. Again, the normal recording we have recorded. After that, we have crescent stimulated. Crescent, it contains, when there is a crescent stimulation, means we are stimulating the post-ganglionic parasympathetic fibers. In case of the human heart, there is a separate innervation by the sympathetic and the parasympathetic nerve fibers. Why there is a regulation? Why there is a supply of the sympathetic and parasympathetic fiber is necessary? As you note that if you stimulate if you remove this part, you cut the vagus fiber in case of the heart, then the intrinsic heart rate, that is a heart rate of the impulse generation by the sinus venusus is about up to 100 beats per minute. And when there is a vagus control over the heart, this vagus control, there is a continued release of the acetylcholine, which will decrease the heart rate from 100 up to the 70 beats per minute. And that continued release of the acetylcholine or the inhibitory effects of the vagus on the heart, that is called as a vagal tone. That is called as a vagal. Is it clear? Now after that, we have added the drug that is a acetylcholine. This acetylcholine has an inhibitory effect. It will act on the nodal tissue. Mechanism that we will discuss with the help of the PPT. And it will slow down the entry of the calcium in the nodal tissue. Ultimately, rate of impulse generation decreases. The pulse of contraction decreases due to, again, it has effects on the cardiac muscle. It will release whatever the calcium entry is there in the cardiac muscle. It slow down the entry of the calcium into the cardiac muscle. As a result, pulse of contraction decreases. So we have seen the both effect decrease. Addition of the acetylcholine decreases the heart rate as well as the force of contraction. Is it clear? After that, we have recorded the normal cardiogram. Again, we have stimulated the vagus nerve or this vagosympathetic trunk so as to identify the site of action of the drug. Is it clear? And it shows again there is a stoppage of the heart in the diastole. Again, we have recorded the normal cardiogram. Again, there is a stimulation of the crescent. Again, there is a heart stops in the diastole, and again we have recorded the normal. Understood? So before and after stimulation of the vagus and the crescent, is it done so as to find out the site of action of the drug? Is it clear? So this is about the acetylcholine. Then we will discuss about the next drug. It is adrenaline, it's also called as a epinephrine. First, we will again record the is a NCG. Again, we will stimulate the vagus nerve. So heart will stops in a vagus stimulation. In your journal, they have given the right vagus stimulation, left vagus stimulation, again crescent stimulation. It's not necessary if you stimulate only the one vagus. Again, the heart will come out from the inhibitory effect. So the normal cardiogram. Here it shows the beneficial effect. Again, there is a crescent stimulation. Heart stops in a diastole. Crescent stimulation. Again, there is a heart will come out from the inhibitory effect. There is a NCG. At this point, we are going to add the drug that is adrenaline. Which is also called as epinephrine. This is the effect of the stimulation of the sympathetic postganglionic fibers. Those sympathetic postganglionic fibers. Now, it has a stimulatory effect. As we have seen concerned with acetylcholine, it is an inhibitory effect, it has a stimulatory effect. So, after addition of this drug, what happens actually? It will opens the number of calcium channels. It will increase the number of calcium channels in the nodal tissue. Is it clear? Ultimately, impulse generation, rate of impulse generation gets increases. Heart rate gets increases. Whatever the conducting tissue is there, it also causes it increases the more number of the calcium channels in the conducting tissues. So ultimately, rate of impulse transport, the traveling rate again increases. It has the effects again also on the cardiac muscle also. It will increases the number of, it will increase the calcium concentration in the cells. Ultimately, it increases the force of contraction. So in this way, this adrenaline will increase the rate as well as the force of contraction. This adrenaline added at this point, then it shows the increase in the rate as well as the height of contraction. So we have to show this in this form. Its effect is increase heart rate, increase force of contraction. Again, so as to find out the, again we will remove this particular point, adding the wrinkle of solution for the heart. Then normal recording will be there. Let's see that will be the NCG. While recording this NCG, you have to take the proportion, height, and number of contraction. Here, after that, we will again stimulate the vagal stimulation. Heart stops in diastole. Again, heart will come out from the inhibitory phase. Slowly and gradually, it shows the staircase phenomena. Again, normal recording of the heart will be there. That is, NCG will be there. Again, there will be stimulation of the crescent. Heart again stops in a diastole. Again, it will come out on the inhibitory effect. And again, shows the normal cardiogram. That will be the NCG. Is it clear? Now, in the examination, no record will be there in this form, only the graph will be there, and they will ask what is the effect of the drug C? So you know that in case of the acetylcholine, effect of the acetylcholine decrease the rate as well as the decrease the heart rate as well as the force of contraction. In case of the adrenaline, heart rate it increases the heart rate as well as the force of contraction. Is it clear? So in the given graph, they will ask you what is the effect of the C. Is it clear? So you will answer directly as a adrenaline, effects of the adrenaline. How you have to find out this is the effect? You have added the adrenaline. Actually, you have to find out first what is the effect actually. What happens in this case? There is increase in the heart rate as well as the force of contraction. Means it is the effect of adrenaline. We have seen the first effect. Then we will find out the drug. Is it clear? So, as in the given graph, there is an increase in the heart rate and the force of contraction has been seen, and it is a due to the effect of the adrenaline. Is it clear? So the given drug will be the it is the effect of the adrenaline. You understood about this? Okay. Now what happens actually in exercise? In case of the hypertension, what happens in the hypertension? There is increase in the blood pressure. Is it clear? Because see, concern with the heart as well as the blood pressure, these receptors for the adrenaline present on the blood vessels as well as on the heart. Then adrenaline will be released. In case of the exercise, what happens? There is an increase in the requirement of the O2 as well as nutrients. But what happens? What will they at this time? There is a release of this adrenaline. This adrenaline will causes it increases the heart rate as well as the force of contraction. It has an effect on the muscles of the blood vessels, that is the smooth muscles. It will causes the dilatation of these smooth muscles so as to increase the blood supply. So in this way, this adrenaline will helpful in the exercise so as to increase so as to increase the blood supply to the native tissue. So this is the effect of this adrenaline in the exercise. Is it clear? You understood about this? Now we discuss the PPT. First part we have seen that we have discussed that is actually this it passed through this part of the ventricle where the actually maximum cardiac muscle is there. Okay, no innervation is there. But you have to take the precaution this should pass through the cardiac muscle only. There should not be puncture of the ventricle. Is it clear? And how this will record this normal cardiogram, what is the systole and what is the diastole that we have discussed. Then we will discuss about the actually how this acetylcholine is synthesized. Actually, we have discussed in the neuromuscular junction, but just to revise it. This occur actually condensation reaction where the acetyl-CoA with the choline in presence of the enzyme choline O-acetyl transferase in presence of the ATP and glucose. This acetylcholine is synthesized, and after synthesis, it is packaged into the synaptic vesicles, where there will occur the release of the synaptic vesicle by the burst of the acetylcholine vesicles. Then the site of acetylcholine release we have seen that is post-ganglionic parasympathetic fibers. We have seen in this research. Post-ganglionic parasympathetic fibers releases acetylcholine. Then pre-ganglionic sympathetic and parasympathetic fibers releases the acetylcholine. Here we have excluded the post-ganglionic sympathetic fibers, which releases the adenine. Is it clear? Then the sympathetic cholinergic nerve fibers, which are supplied to the sweat glands, the ending of this fiber releases this acetylcholine. Then the range of cells in the CNS. When we discuss about the CNS, we will get idea about what is the release of this acetylcholine by the range of cells. Then receptors, particularly muscarinic receptors, and these muscarinic receptors are mainly located on the gland cells, then on the smooth muscle and the heart. Function is when they are present on the smooth muscle and the heart, what happens when there is a release of this acetylcholine? Acetylcholine has a property to, what is its effect? Decrease the rate as well as the force of contraction. Is it clear? So what will be the effect on this heart? Decreases the rate as well. Mechanism that we'll discuss. These nicotinic receptors, these are present on the autonomic ganglion, then adrenal medulla, and motor end plates of the muscle fibers. We have seen that these acetylcholine receptors are called as nicotinic receptors because these receptors are blocked by this nicotine. Then effects. This acetylcholine has an inhibitory effects, and first we will discuss about the generation of the impulse. When it acts on the generation of the impulse, that is a nodal tissue, means that it is concerned with the heart rate. Is it clear? Then it is after generation of the impulse, there is a conduction of the impulse is there. Is it clear? This acetylcholine has all negative effects. All negative effects. So it decreases the generation of the impulse. It decreases the conduction of the impulse. Then it again decreases the excitability of the cells. It again decreases the excitability of the cells because it causes the hyperpolarization. Hyperpolarization means that there is the excess removal of the potassium from the cell to the ECM. And during the stimulation, there is a requirement of the more sodium for the excitation. Is it clear? So in this way, it decreases the excitability of the cells. Then it as we have discussed about the conductivity, the rate of transmission of the impulse. So negative inotropic. Inotropic means nothing but the force of contraction. Then negative chronotropic means the rate of conduction of the impulse. Then negative bathmotropic means there is a decrease in the excitability of the cell. And finally, negative chromotropic means the decrease in the conductivity. You can arrange in this way also rate, conduction, excitability, and force of contraction. What happens first? The rate of generation of the impulse, then conduction of the impulse, and if the impulse reaches up to the cell, then what is the concern with the excitability of the cells? And after excitability, then depends on the contraction of the force of contraction. In this way, we can arrange. Actually, this is a mechanism. If you see this, it has both effects. This acetylcholine site of action is muscarinic receptors in the myocardium. So, in case of this acetylcholine, we have discussed about the acetylcholine, that is a site of action of the receptor, that is a muscarinic receptors. Is it clear? Muscarinic receptors. This acetylcholine decreases the cyclic AMP in the cell, ultimately decreases the cyclic AMP in the cell, slow down the opening of the calcium channels, decrease the calcium concentration, and decrease the force of contraction. Is it clear? As you understood about the requirement of the calcium for the contraction of the muscle in case of the skeletal muscle, smooth muscle, and cardiac muscle. In case of the skeletal muscle, sarcoplasmic reticulum, which is highly developed, which is responsible for the storage of the calcium. but the smooth muscles and the cardiac muscle require the calcium from the external sources. Is it clear? And this system, cyclic AMP, is responsible for the opening and closure of the calcium channel. Is it clear? Then it acts on the cholinergic receptors which are present on the postsynaptic membrane, where it increases the potassium permeability for the cell. What will happen when there is a then occurs the potassium efflux, ultimately hyperpolarization, and heart stops in a diastole. So when you are recorded here, heart stops in a diastole means that it increases the potassium permeability. There occurs the efflux of the potassium, increase the concentration of the potassium in the cells. And when there is a stimulation, again, if you want to stimulate the cell, excess sodium will be required so as to compensate the ionic balance. You have to prepare this mechanism of action. In the examination, you should know how this work actually, how this acetylcholine will work and decrease the rate as well as the force of contraction. How it is removed from the site of action? You know the cholinesterase is present in the synaptic space. Then this cholinesterase will it causes the breakdown of this acetylcholine into the choline as well as the acid. Some amount of the acetylcholine which is present in the synaptic space, it is directly diffused into the presynaptic membrane. Is it clear? And so there is a diffusion from the synaptic space into the presynaptic membrane. Then there is the reacting of the choline, that is utilization of the choline, where actually this choline will be utilized for the again synthesis of the acetylcholine. In this way, this acetylcholine will be destroyed from the site of action. Then what happens? Some drugs are there which will increase the activity of the acetylcholine, or they will inhibit this acetylcholinesterase. What happens? This acetylcholine will act on the receptor for a longer period of time. Is it clear? So it keeps the, if you want to stimulate, if you want to keep the, in case of the, even there is a condition when there is a increase the heart rate, when there is a condition where the heart rate get increased, and if you increase again more and more, then it will causes the pathological condition. There will be the harmful. So we can use such type of the drugs which will inhibit the activity of this acetylcholinesterase. Is it clear? So it will acetylcholine now helpful in this condition so as to decrease the heart rate as well as the force of contraction, and bring this heart rate as well as the force of contraction to the normal. Then some drugs are there. If the acetyl, there is a problem concerned with the release of the acetylcholine. Some drug possesses the property like the acetylcholine. So we can administer such type of the drug and to maintain this decrease. What function is to decrease the heart rate and the force of contraction. One condition we have discussed, when there is a cholinesterase is there, some drugs which will inhibit the activity of the cholinesterase. Here we want to prolong the action of the acetylcholine. But there is a problem concerning the release of the acetylcholine. Now some drugs are there which will work like that of the acetylcholine. So we can use such type of drug in case of the hypertension. These are the drugs actually. Anticholinesterase drug, anticholinesterase drug means that this drug will prolong the action of the acetylcholine. Examples are physostigmine and the neostigmine. And clinical importance is its function is prolong the action of the acetylcholine. In case of the glaucoma, reducing the intraocular tension. What happens in the glaucoma? If the intraocular tension again increases, then it will compress the optic nerve. And when there is a compression of the optic nerve, slowly and gradually there is a decrease in the vision. And if you use such type of the drug, which will helpful for the decrease the tension in the glaucoma. Then I have treatment of the myasthenia gravis. As you know that in case of the myasthenia gravis, what happens? What are the receptors are there? Acetylcholine receptors are there. The acetylcholine receptors get destroyed. A number of these receptors get decreased due to the antibodies which are synthesized by our own bodies. In this case, if there is there should be a proper contraction of the muscle. The acetylcholine action should be continue for a longer period of time. But there are the presence of the cholinesterase, and such type of drug will give in case of the myasthenia gravis, which will inhibit the action of the cholinesterase. And if the acetylcholine is available for a longer period of time, so it is helpful to the contract. There will be the possibility for the contraction of the muscle. Then post-operative paralytic ileus. What happens after surgery? Post-operative paralytic. Post-operative after surgery. Paralytic, concerned with the paralysis. Ileus, nothing but the intestine. After surgery of the intestine, what happens actually? The intestine gets blocked, and it is due to the paralysis of the nerve or the paralysis of the muscle. If you apply such, if you administer such type of the drug, then such paralysis will be, we can remove this paralysis, or there will be a treatment concerned with this paralytic ileus in such a way by prolonging the action of the acetylcholine. Then cholinergic drugs. See the cholinergic drugs, acetylcholine, cholinergic drugs. Drugs which are similar to the acetylcholine. It has an action like that of the acetylcholine. So such a drug name of the drugs are the pilocarpine, methacholine, and neostigmine. Decrease the intraocular tension, same effect will be there as you have seen in abdominal distension and paralytic ileus following surgery. Here what we have done? Here we have inhibited the anticholinesterase. Here the release of the acetylcholine, there is a problem concerned with the release of the acetylcholine. So we have replaced the drug, such type of the drug we can use. Then anticholinergic drugs. Now there is no necessity to stop the decrease the heart rate, or there is not necessary to decrease the course of contraction. So anticholinergic drug, atropine and hematropine, use the in pre-anesthetic medications as well as use the in antispasmodic. Antispasmodic means that spasm of the smooth muscles. If there is a spasm of the smooth muscles, and the name of the drug is antispasmodic, means that they prevents the spasm of the smooth muscles in the blood vessels or the other sides. So those are called as we can use as an antispasmodic. In organophosphorus poisoning, in this poisoning, we can use such type of the drug also. Then this is the graph from the journal. We have seen that this is normal cardiogram. This is vagus stimulation. Again, normal cardiogram has been recorded. This is again recent stimulation, and here the drug is added. ये लेबल नहीं रहेगा. Only this graph will be there on the question. Is it clear? The label गलती से नहीं आया. ऐसा लेबल आने दे. ये रह सकता है. ये फॉरेस्ट है. Then can you observe this graph? ये NCG और यहाँ कंपेयर कर सकते हैं. And what will be your conclusion? In this record, there is a decrease in the decrease in the force of contraction and heart rate. How we have identified there is a decrease in the heart rate? Number of contraction in a centimeter are less as compared to the normal cardiogram. So decrease in the heart rate and force of contraction. Height of contraction is nothing but the force of contraction. Then this is again vagus stimulation, recent stimulation here, and again heart comes to the normal cardio. Which drug is added at A? Name of the added drug is acetylcholine. Then what is the effect of the drug on the heart? Decrease heart rate as well as the force of contraction. What is the mechanism that we have discussed? You should know the mechanism of action of the drug. And last question is, what is the site of action of this drug? Muscarinic receptors. Muscarinic receptors in the myocardium. Then the adrenaline. Adrenaline synthesized from the phenylalanine In presence of the hydroxylase enzyme forms the tyrosine. Again, there is the enzyme hydroxylase. Again, it forms the dihydroxy phenylalanine. Then, in presence of the deoxycarbolase, it forms the dopamine, and it again converts it into the noradrenaline. And in presence of the N-butyl transferase, it can convert it into the adrenaline. Then, this is the synthesis. What are the sites of release? All parasympathetic, all postganglionic sympathetic fibers, all postganglionic sympathetic fibers, except adrenergic nerve ending, adrenergic nerve ending, except postganglionic sympathetic cholinergic fibers. You know that in the adrenal medulla there is a presence of the prolactin cells. This prolactin cells also releases this adrenaline. This prolactin cells at other places, the CNS also again releases this adrenaline. Now the mechanism, receptors are the beta receptors as well as the alpha receptors. These alpha receptors are present on the blood vessels of the skin and the mucosa. Then biliary muscles, again these are present on the sweat glands and smooth muscles of the blood vessels. Again, these beta receptors present on the heart, again present on the bronchial muscles. Now, effect. Adrenaline has a stimulatory effect. Adrenaline has a stimulatory effect on the heart. So whatever the effects we have seen, that is inotropic, it has a positive inotropic effect which will increase the course of contraction. It has again positive chronotropic effects which will increase the rate of contraction. Rate of contraction, that is a rate of generation of the impulse. Positive chronotropic means that it increases the excitability of the cells. Again, positive chronotropic means it is also to increase the conductivity of the conducting system as well as the cardiac muscle. Then mechanism of action. This adrenaline, it again has another name, that is epinephrine. This adrenaline acts on the beta receptors in a nodal tissue, where it increases the cyclic AMP concentration in the nodal tissue, which will open the long-lasting calcium channels and increase the heart rate. Means that firing level, after firing level, it is helpful to increase the cyclic AMP concentration in this. This increase the cyclic AMP concentration, which will increase the number of the calcium channels, and ultimately excess calcium in the cell, which will increase the heart rate. In case of the receptors in the tissue, in the cardiac muscle, it acts on the beta 1 receptors, which will increase the intracellular calcium concentration in the myocardium and increase the course of contraction. In this way, it will increase the heart rate as well as the course of contraction. Then catechol O-methyl transferase. This is the one enzyme which is responsible for the destruction of this adrenaline, and another enzyme, that is a monoamine oxidase, also responsible for the destruction of this adrenaline. Some amount of the adrenaline, if the excess is there, some amount of the adrenaline will be excreted to the kidney. Then the drugs. These are the adrenergic drugs which will has a stimulatory effect. These are the isoprenoline and ephedrine. In the treatment of the bronchial asthma. What is asthma? In anaphylactic shock. What is the anaphylactic shock? This anaphylactic shock means nothing but it develops the allergic reactions immediately if you come in contact with the allergens. In such cases, these types of drugs are used. Then adrenergic blocking drugs are the beta blockers, that is the propranolol and atenolol. Alpha blockers used in the hypertension. In the hypertension, what happens? There is an increase in the heart rate as well as the blood pressure. Means that, so these alpha blockers are used. What is the function of this adrenaline? Adrenaline acts on the alpha receptors and the beta receptors. Is it clear? And what happens? They has a stimulatory effect. Increase the force as well as the rate of contraction. If there is a blocking of this alpha as well as the beta receptor, what will happen? Heart rate will be slowed down and the blood pressure will be slowed down. So the person will be brought to the normal level from the hypertension. Have understood? Then about the beta blockers used in the hypertension, that will be in the angina pectoris and the cardiac arrhythmia. Cardiac arrhythmia is nothing but the irregular beating of the heart. In this case, these are the beta blockers are used, and these beta blockers which will decrease the heart rate as well as the pulse of contraction. In this way, these drugs are used. You should know the names of the drug because in the last examination they have asked question given. List the list of the beta blockers. Question 5. So you should know. So if possible, you can put down these drugs. Yes. Slide series. If I am drawing concern with the I have drawn this graph regarding the acetylcholine on the board. Don't cross this type of the graph in your journal. You have to draw whatever graph I have drawn on the board. Here, difference is there. Here, the decrease in the rate as well as the course of contraction in case of the acetylcholine. Here, we have in case of the adrenaline, we have to show the increase in the rate as well as the course of contraction. You have to draw this graph in your journal. Is it clear? That will be your task for the completion of your journal. Is it clear? Is there any problem concerned with the drawing of the graph? Can you draw this? You have to show the recent stimulation, vagus normal cardiogram is vagus stimulation, then again stoppage of the heart in the diastole, then again staircase phenomena, NCG, then recent stimulation, again this staircase phenomena, NCG, then the added drug, that is adrenaline or the acetylcholine. Is it clear?